Hello, my name is Chen, and I am a PhD candidate in the University of Alabama at Birmingham. Today I'm going to talk about my project, Gene Fusion Detection and Characterization in Long-Read Cancer Transcriptomes with FusionSeeker. We can start with some background about gene fusions. So by definition, gene fusions are the cases when two different genes are fused together in the cancer transcriptome. It is usually a result of genomic rearrangement, such as translocations or deletions, and it is prevalent in a wide array of cancer types, including the prostate cancer, ovarian cancer, and breast cancer. Gene fusions often play critical roles in terms of the tumor genesis and also progression, and they sometimes can serve as the target for the tumor therapy. And previously, we identified gene fusions using the short-read paired end RNA sequencing data, so basically if we observe the two reads from the same read pair are aligned to different genes, and then we can infer the gene fusions from there. However, the short reads, they have a lot of limitations in terms of the gene fusion detection, especially when we want to know which exactly isoform has been fused with the other gene. We can get very little information from the short reads. And as the technologies have developed, we now have long-read RNA sequencing available, such as the HiTSeq from the PacBio and also direct RNA sequencing from Oxford Nanopore. In both cases, we are able to generate full-length transcript sequences that can help us identify the gene fusions with higher accuracy and also to generate the full-length fused transcript sequences for those gene fusion events. And currently, we have two tools that are available for calling gene fusions using the long-read data. And based on our benchmarks, they do have limited performance in terms of fusion detection, especially for those events located in the intronic regions. So here we propose FusionSeeker. It is designed to identify the gene fusion events in both exonic and also intronic regions, and also to reconstruct the fused transcript sequences. Here we have a figure showing the overall workflow for FusionSeeker. If a user has done the long-read RNA sequencing, we do need them to run the MIMAP2 to generate the read alignment file in the BAM format. And we also need the gene annotation file in GTF format as the input so that we can know the location of all the genes. So after we have got both of the input files, the first thing done by FusionSeeker is to identify the raw signals of the gene fusions. And it basically goes through the read alignment file, and for each read alignment, we do the annotation to see if it has been aligned to different genes. And if it does, we do look for two patterns to see whether two genes are located on the same strand or if they are located on two different strands. And in both cases, FusionSeeker will try to infer the breakpoint locations on gene 1 and also on gene 2. And if it fits the pattern for a gene fusion, we do make a record to record the gene name of these two genes and also the chromosomes and then locations and also the read length, read mapping quality, and some other features. So after we have processed the entire BAM file, we will get a large list of the raw signals. And the next step is to cluster those signals into the gene fusion candidates. So the clustering is done really by two steps. The first one is to group those raw signals based on which two genes are fused together. And then we have this density plot density-based clustering algorithm to separate the different breakpoints within the same gene pairs. And this is done by really calculating the distance between between the two raw signals, and the raw signals from the same breakpoint will be clustered together, and the remaining will be discarded as the outliers. And after the clustering process, we do have a filtering process to remove those low confidence events, possibly caused by either errors during the sequencing or errors during the read alignment. And after this step, we'll get a list of the gene fusion events with high confidence. And for each individual one of them, we do perform the transcript sequence reconstruction step, trying to get the accurate sequences. So we will collect all of the reads supporting the gene fusions that also contain the sequence from both genes, and we will perform the partial alignment using the BSN line, trying to get the most accurate consensus sequence, which should have no or very few sequencing errors. that can represent this gene fusion event. So eventually we have the output for FusionSeeker, which will be a list of the gene fusion events together with their transcript sequences. And then the first thing we did is we benchmarked the performance of FusionSeeker in the simulated datasets. So we simulated a human transcriptome with all the normal transcripts plus 150 gene fusions. So we randomly picked 300 genes and assigned them into 100 gene fusions. And for 100 of them, we have the breakpoints located in the exons, and for the remaining 50 events, we have the breakpoints located in the introns. And we simulated both ISOSeq-like reads and also nanopore-like reads based on the simulated transcriptome, and we applied 3 different long-read fusion colors on the simulated datasets. And we did the simulation for 3 times, so all the numbers here are the average number from the 3 replicates. We benchmarked the recall and precision of each fusion color, and we calculated the F1 score based on that. And we see that for both ISOSeq and nanopore datasets, FusionSeeker was able to achieve F1 score over 93%, which was higher than both JFO and also LongGF. And we also looked into what are the events that were reported by FusionSeeker but not by the other 2 tools, and majority of them are actually located in the intronic regions, so this is consistent with the previous statement. And on the simulated dataset, we can further benchmark the accuracy for the transcript sequences. So in both ISOSeq on the left side and also in the nanopore dataset on the right side here, we do see a higher identity comparing to the ground truth transcript sequences that is reported by FusionSeeker consensus sequence, significantly higher than the raw reads, either the ISOSeq reads or than the nanopore reads. And since we have these more accurate transcript sequences, the breakpoint locations we inferred from this sequence is much more accurate than what we can get from the raw reads. So here we further benchmarked the breakpoint accuracy for the 3 fusion colors. We have the histogram here showing the shift of the breakpoints comparing to the ground truth. And for the FusionSeeker, we observed this highest peak in the middle from both ISOSeq and also for the nanopore dataset, which means we have majority of the gene fusions reported with the exact breakpoint locations. And then for the JFO and LongGF, we see that they are more like distributed throughout this entire plot. So that means they have a worse breakpoint accuracy compared to FusionSeeker. So then we moved on to the real data set in the 3 cancer cell lines, the SKBR3 and MCF7 and also HCT116. We picked these 3 cancer cell lines because they have there are some previous studies. They identified the gene fusions using the short-read data and confirmed that they are true using the PCR. And here we run 3 different long-read fusion colors, FusionSeeker, JFO, and LongGF, and we want to see if we are able to report those previously validated events in the long-read data. So here we have the SKBR3, which is the breast cancer cell line. We have the ISOSeq data, and FusionSeeker reported 15 previously validated events, and JFO has 13, and LongGF has 10. And in the MCF7 cell line, we have both ISOSeq and nanopore datasets, and FusionSeeker reported 20 events in both cases. JFO has 23 and then 18 events here, and LongGF has 19 and 20 here. And for the last one, it's a bit tricky. This one only has 2 previously validated events, and all the 3 colors can report 1 of them. So this table kind of shows that FusionSeeker was able to report the most number of the previously validated gene fusions in most cases, except for this one, so that it will have highest sensitivity compared to the previous results among the 3 fusion colors. And we do have some events that are missed by FusionSeeker but somehow detected by JFO and LongGF, and we did look into those events and see what are the reasons that FusionSeeker did not capture them. It turns out that all of those events are supported by only 1 or 2 reads in the long-read dataset, and FusionSeeker was able to identify those raw signals but they were discarded during the clustering process as outliers because too few evidences are there to support them. And there are some features, well, two features I really want to highlight about FusionSeeker in the real data as well. So first thing is that it can identify the breakpoints even in the intronic regions. So this is an example of a previously validated gene fusion event that is present in the MCF7 cell line. And this one is interesting because there are actually two breakpoints located in the first gene here. So the first validated breakpoint is here in the first exon of this gene, and this one is actually reported by all the 3 fusion callers. And we have another breakpoint here in the first intron of this gene. And we can see clearly in the read alignment, we still have many reads supporting this breakpoint. But this one is actually only reported by FusionSeeker, but not by J4 and LongGF. So as this one has been validated through PCR, we believe this is really a true breakpoint, and this can be only detected by FusionSeeker. And in the previous table, for this particular event, they are considered as detected by J4 and LongGF because the first breakpoint is reported. But if we zoom in and be more specific about the breakpoints, then this particular breakpoint is only detected by FusionSeeker itself. And the second feature I want to highlight is about the accurate transcript sequences. So similar in the simulated datasets, we can also compare the accuracy for the FusionSeeker's transcript sequence compared to the raw reads. And this one is compared to the reference genome, so it is more like to be lower estimation of the accuracy because there might be some somatic mutations in there. And in both of the ISMB on the top and also the nanopore datasets, we do see a significantly higher accuracy, base accuracy in the FusionSeeker's transcript sequence than the raw reads. And it's similar here in the nanopore. And also we can get the transcript sequence from the J4's output, but it actually picks any random read that supports this particular gene fusion and use its sequence as a transcript sequence, so that it will come with all the possible sequencing errors in it. And that's why we see similar or a little bit lower accuracy compared to the raw reads for the J4's output. And we can show an example of the sequencing errors here. So this is the IGV plot showing all the alignment for the reads and also for the consensus sequence of FusionSeeker. And here at the bottom, these are the panel for the PacBio's raw reads, ISMB reads. And we see there are a lot of the tiny insertions, tiny deletions, and some of the mismatch here in read alignment. And now this can be observed in the top panel here, which is the FusionSeeker's transcript sequences. And one thing interesting is that we do observe one SNP here. This might be a somatic mutation that is present in this particular exon. And this mutation is actually conserved in the FusionSeeker's transcript sequences, which means in the future, if we have any somatic mutations, they will be maintained by FusionSeeker. And at last, we have some conclusions. So FusionSeeker is able to identify gene fusions with high accuracy in both exonic and also intronic regions, and it can reconstruct highly accurate fused transcript sequences, which can further benefit the breakpoint accuracy. And in the future, we do plan to improve the performance of FusionSeeker so that we can get better performance, especially when we are detecting gene fusions with very low allele frequencies or with very low expression levels. And that is what I have for FusionSeeker, and I really want to thank you for your attention and time.